Hello, Juniata College. My name is Jason Moran. I'm Vice President for Enrollment, and thanks for tuning in for our first feature, Master's Program Monday. Going forward, every Monday at noon, we're going to spend some time highlighting one of our master's programs. In case you didn't know, we have at Juniata College 5 master's programs, beginning with Master's in Bioinformatics, Master's in Data Science, a Master's in Business Administration, Master's in Organizational Leadership, and also a Master's of Accounting. As we go forward here, each week going forward, we're going to spend some time featuring each of those programs, having the opportunity to meet our faculty, highlight student work, and also why these programs, why Juniata College. Beginning today, we're starting with Bioinformatics. I'd like to bring in Dr. Gina Lamendella, one of our professors at Juniata College, who also directs our Bioinformatics program. And Dr. Lamendella, we'd love to hear a little bit about you, a little bit about our Bioinformatics program, and why should students pursue bioinformatics, and what can students do with a master's degree in bioinformatics?
Absolutely. Thanks for having me, Jason.
Sure.
So this is my 10th year at Juniata College. I'm coming to you from Berkeley National Lab. That's actually where I grew my passion and love for bioinformatics. So very quickly, early on in my graduate days, I fell in love with microbes. So I'm a molecular microbiologist and also a bioinformaticist. We're going to talk a little bit about what bioinformatics is in a bit. But when I was doing my graduate degree, essentially I was working for the Environmental Protection Agency, and my goal was to actually track sources of fecal contamination in surface waters. And the way I did that was by tracking different microbes that are associated with different types of fecal contamination. So I generated a lot of data, a lot of DNA sequence data, and I was good at generating all that data. But back then, there weren't really bioinformatics platforms, there weren't bioinformatics programs, and so I had to sort of develop things on the fly. So after my graduate school, I said, well, heck, I better get more training in computer science and IT and learning how to analyze all these data. So I went to the Joint Genome Institute, which you may know, that's where the first human genome was sequenced. So I learned from the best of the best there about how to analyze these really large, complex data sets. And that's what bioinformatics is all about. It's really about leveraging computer-based technologies to analyze and understand biological processes. And this could be anything from sequencing a genome all the way to discovering mutations and what mutations might lead to types of cancers. Bioinformatics is also developing and discovering drugs. Pharmacogenomics is a huge topical area, so there's a lot of different applications of bioinformatics, and it's highly interdisciplinary. So our program is generally based around getting you guys used to and using these really complex tools and learning how to apply things like statistics, mathematics, computer science, and the basics of bioinformatics in your core courses. And then in your elective courses, you can actually shape what type of bioinformatics you want to do. So you may want to do medical informatics and work for a hospital, right, and learn how electronic medical record systems work. You may want to do drug discovery and learn how proteins work and interactions. You may want to discover things like mutations and, again, how those might be leading to things like cancer. So.
Excellent. And so what I know is so important for a student and their curricular journey, specifically looking at bioinformatics, are the facilities and the equipment that they get to use to be trained in working with this equipment after they finish their degree and they enter into the world and into the workplace. And so tell us a little bit about where are we and some of the equipment that we're going to feature today.
Yes, absolutely. So we are in CSI, which stands for Contamination Source Identification. This is a company that I started with 2 Juniata alums, Justin Wright and Gary Shope. And we are changing the gold standard for infectious disease diagnostics. So we are trying to figure out how to get physicians more accurate information about what pathogen is causing a specific disease, really specifically and quickly. And so that's what we're doing here. So we are in CSI. We have about 4,000 square feet of space here. And I'm going to take you on a tour in a little bit. We're going to step you through the process of how we do this revolutionary disease diagnostics. And so it's actually a very simple idea, very simple, elegant idea, in that essentially we take a sample and we break open all the organisms in that sample, and then we sequence their DNA. And you can think of that DNA, Jason, as a little thumbprint,
Sure.
right, for identifying who's there. So just like you have a thumbprint, I have a thumbprint that identifies us, these microbes have DNA, which is like a little barcode for telling us who's there. This is a very revolutionary way of trying to identify these different organisms and what organisms might be causing disease. So you want me to take you through the process?
Would love to see it.
That'd be great. So essentially there's sort of 5 main steps, right? We acquire a sample, and that sample could be anything from a blood sample to a urine specimen, synovial fluid, what have you, any sort of human associated sample. And then that sample needs to be accessioned. So we need to bring it into our system so that we could forever track it through the laboratory steps and through the bioinformatics analysis, all the way through actually sending that report to the physician. So all of that, it's sort of, you can think of CSI as a one-stop shop for doing all that. And CSI is actually, we have over 20 employees, a majority of which are current Juniata students, undergrads, master students, and recent alum. And they have partnered with us over the past 2 to 3 years, developing patent-pending wet lab and bioinformatics procedures.
Okay, excellent. So, you're going to walk us around and show us what
Yes.
we're working on?
Absolutely. So I'd like to introduce to you Justin Wright.
Excellent. How are you, Justin?
Hi, Jason. How are you doing?
Good, doing well. Thank
Hello,
you.
everybody.
So Justin was one of my very first research students when I was a young professor here at Juniata College. And Justin was going to go to med school, but decided that he wanted to do research instead.
Yep.
And so I had the pleasure of meeting Justin his senior year. And that's when we started this idea of starting a research-based company. Justin's also one of our master's students in the bioinformatics program.
Yep.
And he's going to step you through probably the most important part of this process, which is accessioning the sample. So you can imagine during the height of COVID, so our laboratory here pivoted from what we were doing, all of our research and development, to help with the COVID crisis. We served over 20 different nursing homes, hospitals, schools in the area to help keep them safe. And the process really started with Justin developing this lab information monitoring system to track a sample all the way from us receiving it, again, all the way through generating that report and getting the answer to the physician. And because of the platform he developed, we were able to have an average turnaround time of less than 24 hours and helped especially the nursing homes. This was really a really neat story in that nursing homes, the COVID was spreading quite quickly. We know it was afflicting the elderly disproportionately, and we were able to help 2 or 3 nursing homes prevent spread and outbreak of COVID within their facilities. So I'm going to turn it over to Justin to nerd out a little bit on this LIMS
Sure.
platform that he developed with other graduate students and undergraduate students.
Absolutely, yeah. Always happy to nerd out. So, yeah, I think something really important that Gina alluded to in her presentation of the importance and relevance of bioinformatics is the fact that there's so many different applications that one can contribute to the field. So I think the kind of traditional understanding of bioinformatics is, data analysis, data processing, churning of large data sets, which is very relevant and something that we work on frequently at CSI. But on kind of more of the back end, a very important component of bioinformatics and computational biology as a whole is the development of systems that facilitate efficient processing of biological specimens. So at CSI, we've not only invested in our own proprietary data analysis pipelines, but we've also invested a tremendous amount of time and effort into building what's called our LIMS system, our Lab Information Management System. So something that Gina alluded to, one of the most common reasons that clinical labs get what's called essentially a demerit from the Department of Health is that samples are lost or misassociated with the wrong patients. So we knew that we had to invest in a cutting-edge LIMS platform that allowed us to track a sample from the beginning of its life cycle when it arrives to our lab to when we report out on the results to clinicians. And we were really put to the test. No one was expecting, obviously, the COVID-19 pandemic, so we knew we had to create something that could facilitate very high volume of sample processing. So we processed over 50,000 COVID-19 samples over the course of the pandemic, And we actually get a little report card at the end of the year from the Department of Health that showcases how many, how timely were we and how accurate were we in delivering the correct results to the correct patients. And we actually had 100% accuracy of all 50,000 reports that were delivered to our clinical collaborators. We were able to get all of those samples in timely and effectively. So I unfortunately don't have, well, I guess it's a good thing that I don't have any COVID-19 samples to process. But essentially it works kind of like you'd expect when you see in the movies. You basically, every sample we get comes barcoded. It's simply scanned into our system. All of our accessioning is electronic. And therefore, as soon as we scan that sample into our LIMS system, the associated order is pulled up. You'd be surprised, the majority of clinical labs out there right now actually utilize paper order forms, which you can imagine adds a lot of processing time. So on the informatics knowledge side of the fence, we've kind of invested our time and energy into building these systems to keep things moving quickly. So as soon as we scan a sample in, like that, the sample is then recognized, it's QC'd by one of many accessioning personnel, many of which are Juniata alumni. And then if the sample passes a physical inspection, it's then sent to the main lab for processing. And we have a bunch of cool technology. This is just the very beginning of what we do at CSI and how you can apply your informatics career. But once QC is assessed, we then go to the fun part, which is actually processing the sample.
And is there a different location you want to take us to show us where the processing of the sample takes
Yes,
place?
let's go to the lab where the molecular magic happens.
Awesome. All right, so now we're in the place where the molecular magic happens. So Dr. Lamondello, Justin, tell us where we are.
Welcome to the main lab. This is where all of the COVID processing occurs. So this is where all the molecular biology steps happen. So we do the conventional PCR-based COVID testing that tells us whether a patient was COVID positive or COVID negative. And those samples are processed under the hood behind us by lab testing personnel, again, many of which are current graduate students, a recent alum of Juniata College. And then the other thing I really wanted to talk to you about was our proprietary technology called CSI-DX. And this technology is a patent-pending technology that we've developed with our graduate students, with Justin and the team, where we're actually able to detect how COVID is changing and mutating over time.
Okay.
And this is really important, right? We know that COVID is an RNA virus and it's constantly changing. It's learning from us, it's getting better at infecting us, it's getting better at spreading. And so we want to track those little DNA or RNA mutations over time. And so basically what we do on the robot behind me is we break open a specimen. So the specimen comes in, we break open the cells, and if there's SARS-CoV-2 in there, we're breaking open that viral capsid as well. So you can think of this virus as a protein shell with RNA inside, and on the outside of it, there's all these little spike proteins that you may have heard of.
Yep.
Those little spike proteins are the ones that enable the virus to attach to and infect our cells. And so using our technology, essentially what the robot's doing behind us, is it's extracting the RNA from the virus and preparing it for RNA sequencing. And so by decoding the sequence, remember RNA is made up of 4 different little chemicals, by decoding the pattern of that sequence, we're able to track over time how the genome is changing, how that spike protein might be mutated. And so all of that's occurring on the machine behind us.
Excellent. So, a lot of other features that you could show us throughout the lab space. And if you want to check out those features, for those of you tuning in, you'll have to check out a future Master's Program Monday upcoming feature. Question I do have, interested in a Master's in Bioinformatics, what are you looking for in an applicant?
We are looking for applicants, our applicants come from all different disciplines. So I have had students right out of undergrad from biology, chemistry, computer science, mathematics. And the great thing is you don't have to have, just like I didn't have any computer science knowledge before I embarked on bioinformatics, you don't have to have that much background in computer science. So whatever level you are at, as long as you have some basic biology and basic chemistry courses under your hat, you will succeed in this program. So we have had undergrads, their 4th year start the program and finish it in as soon as a year. We've had adult learners come back after being in the field for 10 or 15 years, succeed in the program. And our students are doing wonderful things. I think we've had over 10 graduates so far, some of which who are now getting into the PhD programs that they couldn't get into upon their first shot, but after their master's degree, they've gotten into these places. We have people working for top 50 biotech companies, doing things like drug development, biotechnology development. So.
Excellent. So if you want to learn more, check us out on the web, Juniata College, academics, specifically graduate programs in bioinformatics to learn more. We host daily information sessions. Also, if you want to come and visit and see the space, feel free to set up a visit with me. You can find my information right on the web on Juniata College Graduate Programs website. Tune in next week, same time, same place, Master's Program Monday at noon. We'll be featuring our Master's in Business Administration, or the MBA program. But thank you, Dr. Lamondello. Justin, thank you for giving us a tour and talking a little bit about the program. Until then, see you later, Juniata College.